In Module 1.1, we are going to learn about the experimental techniques in cell biology. I have confined the topics to mainly cover some of the routine methods used by scientists to study the cells. The topics will include growing cells in the laboratory outside a living animal, visualization of the cell and its structural components, and isolation of subcellular components. By subcellular components, we mean the components that make up a typical cell. In addition to the topics that we will discuss, there is a vast area of biochemical and molecular genetic techniques that are used in cell biology, but that is beyond the scope of the current course. You have learned some of these techniques in your biochemistry prerequisite, and we will refer to them often throughout the course. So if necessary, please review these topics mentioned in your syllabus. Some of the molecular genetic techniques you will learn in other courses like genetics and molecular biology, addressing exclusively gene and protein functions. These techniques are beyond the scope of this course, and we will not discuss those. First, we are going to talk about how we can grow cells in the laboratory. Why do we want to grow cells in culture? To have a molecular, biochemical, and genetic understanding of cells require large population of cells that are homogeneous. One way to do this is to purify large population of cells from tissues and then conduct biochemical studies and genetic analysis. But if you are working with a very rare cell type, then it is inconvenient to purify cells from a very large volume of tissues, plus it is expensive and labor intensive. So it is often easier to isolate the cells from tissues and propagate or multiply them in culture. The first step for isolating cells from a tissue is to disrupt the intercellular interactions that hold the cells together in the tissue. There are several commercial disruptive agents, but there are two broad classes of disruption agents, and you will probably need to use both. You usually treat the tissues with a protease such as a collagenase or trypsin that will hydrolyze or cleave the protein-to-protein interactions between the cells and between the cells and the extracellular matrix. Cell-to-cell and cell matrix interactions rely on calcium as well. So you can use calcium chelators like EDTA to disrupt the intercellular attachment and facilitate the release of cells from the tissues. After this first step, you have a heterogeneous population of cells, and usually not very helpful if you're interested in one cell type. Most cells, given the appropriate conditions, can be cultured in the lab. These conditions include growing cells in a rich medium, usually consisting of at least the essential 9 amino acids, some vitamins and peptides and protein growth factor, which are primarily supplied by the addition of some type of serum, usually fetal calf or bovine serum. Most cells prefer to grow on a negatively charged solid surface, which mimics the extracellular interaction. So the dishes and flasks that the cells grow on are negatively charged, most always a type of plastic. When cells are propagated directly from the tissue of an organism, they are known as primary cells. Some examples of primary cells are fibroblasts, skeletal muscle cells, and nerve cells, which can be isolated directly from an organism and cultured in the laboratory. One of the advantages of the cultured cells is that they display all the characteristics of the tissue from which they were isolated. So for an example, primary fibroblast will secrete collagen, skeletal muscle cell will fuse spontaneously, contract, the nerve cell will form synapses and communicate with each other electrically. The disadvantage of the primary cell culture is that the cells have a finite doubling somewhere between 50 and 100 times in the cell culture. There are some terms that we use in the cell culture. A cell strain is defined as a single cell lineage from one initial primary culture. Suppose we isolated a tiny portion of intestinal tissue by biopsy, break it up mechanically, and then digest it with trypsin, collagenase, and EDTA so that the cells come loose from the tissue. We then collect the cells and continue to establish a primary culture. All cells that will be derived from this initial primary culture by division will be the same cell strain. Now, as we grow cells in culture, the cells will grow and cover the whole surface of the culture vessel. This is called confluency. The primary cell culture will stop growing when they become confluent, which is known as contact inhibition. So at this point, if we do not split the cells into new culture vessel, they will eventually die. So this splitting of the cells into new culture vessels with a fresh media is known as passaging the cells. One interesting phenomenon we are going to observe later that immortal cells or cancerous cells often do not exhibit contact inhibition like the mortal primary cells in culture, and can grow on top of each other and often in suspension or floating in the medium without even attaching to the surface of the vessel. We will revisit this in the last module of this course when we will study cancer cell biology. This figure shows the growth stages of the cells in culture. On the y-axis is the growth rate, and the number of cell generation is shown on the x-axis. So freshly isolated primary cells have a rapid growth rate, but about 10 to 12 generations, the growth rate begins to level off, and by 50 generations, you can see that there is a significant amount of cell death. So you can really only study these cells while they are actively growing off in phase 2. But by 50 generations into phase 3, these cells begin to die and show characteristics that are abnormal, means often shows abnormal phenotype due to accumulated mutations. Most of these mutations are lethal and leading to gradual death of the cells. But sometimes several combinations of mutations can change the phenotype of these cells to what we called are transformed cells, and they start to grow back again. This is the phenotype we often observe in cancer cells, and we will talk about that in the next slide. As we have seen in the primary cells have infinite lifespan, one way to circumvent this problem of finite doubling is to use transformed cells, the concept we introduced in the last slide. So the transformed cells are usually derived from a tumor or from cells that have undergone spontaneous genetic mutations. Sometimes this is called oncogenic transformation, which makes these cells immortal. They do not have a finite doubling and can grow to very high densities and not necessarily even on solid surfaces. The disadvantage is that they are genetically altered. They may not accurately represent the original cells in tissues and may display abnormal properties which could skew your conclusions about certain cellular functions. I want to talk about culturing embryonic stem cells. They have very fastidious growth requirements as compared to other cells. This is how the embryonic stem cells are obtained. If you want a human or mouse stem cells, you start with a cleavage state embryo which is developed by in vitro fertilization. The embryo is then cultured in the lab to the blastocyst stage. Within the blastocyst is the inner cell mass, which contains the embryonic stem cells. So the inner cell mass is isolated and placed into the culture medium. Some embryonic stem cells grow best on a layer of fibroblast feeder cells. So rich medium is not enough. They have to be in contact with other cells. However, it has been shown that some embryonic stem cells can grow well without the feeders if certain specific cytokines are added to the media. So the fibroblast feeder cells are not needed. So, two controversial issues with embryonic stem cells are: first, when the inner cell mass is isolated, it destroys the blastocyst. This makes embryonic stem cell research controversial because you are destroying an embryo. The other issue is the use of the feeder cells. When you are growing the embryonic stem cells on the feeder cells and then start measuring or quantifying certain physiological characteristic, the question arises, are you measuring the physiological characteristic of the feeder cells, the embryonic stem cells, or a combination of both? This problem is solved somewhat with the use of the cytokine instead of the feeder cells. And at confluency, the embryonic stem cells outnumber the feeder by almost 20:1. So it's not really a big issue. But again, this has been a problem in the embryonic stem cell research. Later on in the course, we will talk about the cell cycle and talk more about embryonic stem cells, but I wanted to bring them up this point to mention that they do have more fastidious growth requirements and are more difficult to grow in culture than the primary or transformed cell lines. So you need to exploit some cell property in order to purify your cell type of interest from this heterogeneous cell population. You can use physical properties such as density. Red and white blood cells have very different densities, so you can use centrifugation to spin out the cells you are interested in. You can use physiological property. For instance, if you have a mixed population of cells and within that population you have macrophages. The macrophages have an affinity for glass, so you take the mixed population of cells and expose them to a glass surface, allow the macrophages to adhere, and you can wash off all the other cells. This results in a fairly good efficient purification of macrophages. Flow cytometry takes the advantage of proteins that are on the surface of the cells of interest. For example, say you are interested in studying the T cells from the spleen. The T cells have on their surface two proteins, say Thi1 and CD3. These two surface proteins are unique to the T cells and are not found on the surface of other cells of the spleen. So flow cytometry exploits this uniqueness to isolate T cells from this heterogeneous cell population of the spleen. The first step is to homogenize the spleen using disrupting agents such as proteases, we talked about it, like collagenase and trypsin, and calcium chelators like EDTA, and you end up with the heterogeneous population of cells. Then you use a fluorescent antibody that specifically bind to Thi1.2 and CD3. In this case, this is a red fluorescence conjugated anti-Thi1.2 antibody and a green fluorescence conjugated anti-CD3 antibody. So you stain your cells with the fluorescent antibodies, and only the T cells will bind to the antibodies and become fluorescent. So then you use a flow cytometer to separate those antibody-bound fluorescent T cells from all other cell types in the mixed population obtained from the spleen. This is a diagram of the setup of a flow cytometer, cell sorter, or fluorescence activated cell sorter, or FACS. It is a very elegant system. You take the fluorescent stained cells that are in suspension in a liquid known as the sheath fluid. The cells are fed into the cytometer through a sip which allows for cells to pass through the laser light beam as single cell. There are two detectors on this system. One is a scatter light detector which detects the cell based on its size and granularity. The second detector is a fluorescent light detector which identifies, in this case, the fluorescently labeled T cells. So as the cell passes by the laser, the fluorophores are excited. The suspension is atomized into drops, and the flow cytometer will detect the drops that actually have cells in them. Once the flow cytometer detects that a drop has a cell in it and is fluorescing, it puts a negative charge on the drop. The drops then pass between two polarized charge plates. One plate is negatively charged and another plate is positively charged. The drops with no charge, so no cell or no fluorescing cell, would be collected in this collection tube and be discarded. But any drops with a charge on it, which mean it contains a cell labeled with a fluorescent antibody, that drop will be deflected by the charged plates and be collected. Then you have the T cells separated from the heterogeneous population, and you can take those T cells and study their biochemistry, genetics, and molecular characteristic. This is a dot plot from the flow cytometer showing the separated T cell population. Along the x-axis is the red fluorescence, and along the y-axis is the green fluorescence. And in the lower left quadrant is a large population of cells that are unstained or have very low fluorescence and are non-T cells. In the upper right quadrant shows strong green as well as red fluorescence, indicating the fluorescent stained T cells. Please note that in flow cytometry, there is a convention of numbering each of these quadrants. So here in the red, quadrant 1 is designated as Q1, quadrant 2 as Q2, and so on. Some cells, like epithelial cells, say for example, that are found in the intestinal lining or lining of the kidney tubules, have a polar nature. What we mean by the polar nature is that these cells have an apical surface or top. This top or the apical surface face the hollow or lumen of the tube being either your gut or say the kidney tubule. This apical surface of the cells are free from any obstruction and can sense all the material that are passing through the lumen or the hollow of the tube, bumping and rolling randomly on the apical surface of these cells randomly. In contrast, these cells also have a lateral surface that are attached to the next cells and are not free in that sense. In addition, the basal surface, or the exactly opposite surface of the apical side, is responsible for attachment or anchor these cells on a membrane called basal lamina or basal membrane of the tissue. So you can see that there is a polarity in these cells. The apical pole is free towards the hollow of the tube, sense material passing by, and can absorb material or secrete material to the lumen of the tube. These includes a variety of ions, other organic materials, say sugar or amino acids. On the other hand, the basal pole perform a different set of functions, which include attachment and often some other kind of transport mechanisms that we are going to discuss elsewhere. But the take-home message is that they are different on each pole, and hence they have a polarity. In contrast, think about your blood cells freely floating through the blood do not have any polarity. So to culture these polar cells, a 2D and a 3D culture system have been developed that can mimic the tissue space in the body like this. In this above picture, this MDCK, which is Madden Derby canine kidney or a dog kidney cells, are being grown on a specialized container in a monolayer or single layer. This yellow apical media mimics the hollow of the lumen of the kidney tubules for the MDCK cells, as they are kidney cells. The lateral surface of the cells are attached to the cells next to it by cell adhesion molecules or CAMs, and this attachment requires calcium in the media, which is provided. The basal surface is resting on a porous filter which acts as a basal lamina for the attachment of the basal surface of the cell. Note that the media color in the basal media is blue compared to the yellow apical medium, which denotes the difference in the composition of these two media as observed in the real tissue space in the animal body. The media may vary in pH, ion composition, nutrient composition, etc. So you can see that the 2D and the 3D culture system can mimic the tissue space to grow polarized cells that require different environment in different part, that is the apical part and the basal part of the cell. So here you can see now a 3D printer are being used to make a pliable 3D matrix to grow cells, and there are some success of even growing organ-like tissues in the cultures known as organoid culture. The bottom panel shows a cyst of MDCK cells grown in this 3D cell matrix. The left side shows an unstained bright field microscopic image of the cyst that is not providing much information. But on the right-hand side, this is an image of the fluorescent microscopy. The cells have been stained with a red dye or a fluorophore, which is specifically will only bind to a protein found on the apical surface of the cell. So you can see the apical surface is stained red facing the hollow or the lumen of the structure. On the other hand, the green dye or the fluorophore is found on the lateral and the basal surface only and not on the apical surface. And you can see the distribution on the lateral side of the cells and the outer periphery, which is the basal surface of the tube in a cross section. The nucleus are stained in a blue with a DNA-specific dye, by the way. So let me first introduce the concept, as many may not have the immunology background to understand what is being done and why. So what are monoclonal antibodies and why we need them? Let's first define some terms. When a vertebrate body like ours encounters a foreign material that is not found in our body, is known as an antigen. The antigens are predominantly proteins or peptides, but can be combination of proteins, carbohydrates, lipids, nucleic acid, even a variety of inorganic and organic materials. Some of these antigens are immunogenic, but not all of them. Means when the immune system cells see these foreign antigen, they will trigger a cascade of reactions, and if the antigen is associated with a pathogen, the body will produce a kind of a molecule that is known as an antibody. These antibodies are Y-shaped molecules, usually made up of 4 protein chains, and each of them can recognize an epitope of an antigen. So what is an epitope? Let's think this way, that you have an antigen known as protein X. All proteins have a distinct 3D structure and may have many 3D features that can be recognized by the immune cells. For an example, say the protein X has a purple, red, green, and a blue epitope, or distinct 3D features as shown in the bottom inset. In reality, often much more epitopes are present on a protein though. Now, when this foreign antigen protein X enters the body, each epitope will be recognized by a different immune cell, and for each epitope, a different antibody will be produced by the B cells or B lymphocytes. Say for an example, you will have an anti-violet, anti-green, anti-red, anti-blue antibodies. So this collection of antibodies against one antigen, protein X, is known as polyclonal antibodies. For most diseases, pathogen invasions, this is a powerful method as several antibodies bind to the antigens and neutralize them. But for many biomedical needs, such as labeling a specific cell type, targeted cancer therapy, or many types of diagnostic, we require an antibody against a single epitope of a protein. That is known as a means single, monoclonal antibody. For example, in many cancer cells, a protein is mutated to form a different or abnormal epitope. These targets are used to raise monoclonal antibodies for diagnostic as well as therapeutics. So how do we do this? Suppose we are trying to raise a monoclonal antibody against the red epitope of the protein X, which is a human protein. So we will purify the protein from the human cells and inject it into a mouse. Now remember, the protein X is a human protein, so it is a foreign antigen to the mouse. Now the mouse B cells will produce lots of polyclonal antibodies against all the epitopes that is present in the protein X. But a specific B cell clone will produce only the antibody against the red epitope. So this is the specific monoclonal antibody produced by one of the B cells or lymphocytes in the mouse that we are interested in. So specific monoclonal antibodies are difficult to isolate from polyclonal antibody mixtures, but monoclonal antibodies can be produced by cultured hybridoma cells generated by fusing the B cells with an immortal myeloma cells. To do this, immortal myeloma cells, which are the cancerous transformed cells, are fused with the normal antibody-producing spleen cells, B cells of course, by polyethylene glycol or by Sendai virus. They are used as fusing agents. The myeloma cells that are used in this method is unable to synthesize purines or precursor of nucleotides to synthesize DNA by an alternative pathway, as they lack thymidine kinase, TK, or AGPRT, hypoxanthine guanine phosphoribosyl transferase enzymes. The myeloma cells used here must be lacking either AGPRT or TK or even both, but at least one of those. But the myeloma cells can use a de novo or savage pathway, which is separate from the alternative pathway to synthesize the nucleotide precursor. So, subsequently, we will see the use of a media known as a HAT or hypoxanthine aminopterin thymidine media. This is a selection media which blocks the de novo or the savage pathway. But the mouse B cells have both TK and AGPRT in them. So now we have a potentially fused myeloma cells with the B cells, and they are not put into the HAT selection media. The unfused B cells will die even if they have the TK or AGPRT, as they are mortal and have limited lifespan. The unfused myeloma cells will also die as they lack the TK or AGPRT, and the media contain a HAT which blocks the de novo or the savage pathway to synthesize the purines or precursor of nucleotide for DNA synthesis. So there is no pathway is being allowed by the myeloma cells to synthesize DNA. So de novo, savage pathway, alternative pathway, all are blocked for these unfused myeloma cells. But only the fused B cells with the myeloma cells will survive, as they have the TK and AGPRT from the B cells and will be able to use the alternative pathway to synthesize the purines, and will also have the immortality coming from the myeloma cells. Now, each of these fused cells are known as hybridomas, are separated in single wells in the cell culture, and tested for the monoclonal antibody against the red epitope. That specific hybridoma cell clone producing the specific monoclonal antibody is now immortal and will be permanent source for the production of the monoclonal antibody in research labs or industry, and can even be distributed in different laboratories. Hypoxanthine aminopterin thymidine media or HAT medium is a selection media for mammalian cell culture, and let's elaborate on that, which relies on the combination of aminopterin, a drug that acts as a powerful folate metabolism inhibitor by inhibiting the enzyme dihydrofolate reductase, which produce hypoxanthine, a purine derivative, and thymidine, a deoxynucleoside, which is a precursor of deoxynucleotide or building block of DNA, which are intermediate in DNA synthesis. The trick is that the aminopterin blocks the DNA de novo synthesis, which is absolutely required for cell division to proceed. But hypoxanthine and thymidine provide the cells with the raw material to evade the blockage, which is the de novo or the savage pathway of nucleotide synthesis, by an alternative pathway provided that they have the right enzyme, which means have functioning copies of the genes that encode them. And these are the AGPRT and TK. So the two genes that help the cells to use the alternative pathway to bypass the savage pathway are either AGPRT, hypoxanthine guanine phosphoribosyl transferase, or TK, thymidine kinase. So the mortal splenic B cells are positive for thymidine kinase, they are TK plus, are fused with either TK negative or AGPRT negative myeloma cells, which are referred to as plasmacytoma cells in the middle panel of the figure. The fused cells are incubated in the HAT medium. The aminopterin in the medium blocks the de novo savage pathway of nucleotide synthesis. The unfused B cells can produce TK or AGPRT and use either de novo savage or alternative pathway to synthesize the precursor of the nucleotide. So even in the HAT media, as they can use the AGPRT or the TK, they can survive. But they will not, as they will die naturally, as they have a short lifespan and are not immortal. In this way, the only B-cell myeloma fusion hybrids have the immortality of the myeloma cell and the genes that synthesize AGPRT or TK from the mortal B cells. The resultant fused hybrid is known as hybridomas, now become immortal and can use the alternative pathway to produce the nucleotide precursor. These hybridoma cells will produce the monoclonal antibodies that will be secreted in the cell culture medium and can be purified. Once established, each hybridoma cell cone will produce an antibody against a single epitope, and hence they are called the monoclonal antibodies. It is not always possible to find a patient with a particular disease, and practically and ethically it is impossible to study diseases in a patient's routinely except a few instances. So the scientists have to study them in vertebrate animal models, but even in the animal model studies have limitations. But as many human diseases are associated with specific cell components and their malfunctioning, studying cells in culture provide an extremely powerful tool to study many diseases. For an example, receptor-mediated endocytosis was elucidated from hypercholesterolemia patients. What I want to focus on are some of the drugs that are commonly used in cell biology. There is a large number of commercially available compounds that can be used to inhibit the function of organelles or function of specific proteins within organelles. The drugs that I have listed here are some of the very common ones, and we will mention all of these throughout the course in different modules. For example, a very common compound is PMSF is used in the media whenever you break up some cells to isolate organelles, proteins, or nucleic acid, or do other biochemical assays. The reason being the cells are loaded with these serine proteases, and as soon as you break open a cell, the vesicle cell lysosome containing them will also break. And these proteases will degrade many proteins quickly. To prevent the protein degradation, PMSF is routinely used with many other protease inhibitor as well. We will mention albumin in the context of transportation of small molecule like sodium and potassium across the plasma membrane. Then we will talk about tunicamycin and brifaldin A in the context of protein glycosylation, trafficking, and secretion. Phalloidin, cytochalasin, latrencholine, Taxol, colchicine, noconazole, all will become relevant while studying the cytoskeletons. We will mention the forskolin, how it can activate adenylate cyclase and modulate signal transduction. Monoastrol is a drug that inhibits a motor protein known as kinesin 5 associated with microtubules. We will talk about this in detail when we will study the cytoskeletons. In brief, the kinesin 5 motor is necessary to push the spindle poles apart to form a mitotic spindle that is needed for the chromosomal segregation during the cell division or in the mitosis. The drug monoastrol inhibits the kinesin 5 motor protein and the mitotic spindle cannot be formed. This process will halt the cell cycle and the cell will stop dividing. This is why the drug is being tested in certain tumors to halt cell division. You can see in the figure, the upper one has a perfectly developed mitotic spindle, the greens are the microtubules, and the blues are the chromosomes lined up on the microtubules. In the bottom figure, in presence of the monoastrol, the mitotic spindle did not form, or the spindle poles could not separate and remained aggregated in one area. This concludes the part 1 of the module 1.